Hi students, in this part we're going to start covering chapter 6, and these are the topics to be covered in this chapter. So in this recording I'm going to cover section A, which would be talking about the cells of the nervous system. So we'll talk about the structure of a neuron, and then we'll talk about the classification of neurons according to function and according to structure. We'll talk about the glial cells, and then what would happen if a nerve is injured? Would it regenerate or not? Okay. The other sections will be discussed in other videos. So the major two divisions of our nervous system, structure-wise, so according to anatomy, would either be the CNS, and that would be the different parts of the brain and the spinal cord, or the peripheral nervous system, and that would be made out of the nerves that come out of the brain or the spinal cord. So the nerves that come out of the brain are known as cranial nerves, while the nerves that come out of the spine are known as spinal nerves. And then those nerves are going to innervate structures. Those structures could be muscles, could be glands, or it could be taking senses from different organs like the ear and the eye and so on. The functional unit of the nervous system is known as a neuron. The neuron is also known as the nerve cell, so we can use those interchangeably. Now, this is what a typical neuron looks like. When we get to the anatomical or the structural classification of neurons, you'll see that we have different kinds of these neurons, but the majority of the neurons look like this. We got a cell body, and the cell body has the nucleus, and then there are dendrites that bring information into the cell body. Then there's an axon hillock. The axon hillock looks like a funnel-shaped area that would funnel the information or the electricity, also known as the action potential, to go down the axon all the way down to the axon terminal. The axon terminal is what connects to either a muscle or a gland or so on. So again, for the direction of the action potential or the direction of electricity, it would go from dendrite into the cell body, and then from cell body down the axon, away from the cell body towards the axon terminals. This axon hillock or this funnel-shaped area is extremely important, so that when we get to talk about the electrical changes that happen on the axon, all of these changes they start at that axon hillock. So what about the myelin sheath? Myelin sheath, you want to think of nerves or axons kind of like wires that need to be insulated from the rest of the body, just like when we have electrical wires covered with insulation. The insulation in our body is known as a myelin sheath, and myelin sheath is made either by Schwann cells or oligodendrocytes, depending on where that nerve is. So if the nerve is in the peripheral nervous system, the myelin sheath would be made by Schwann cells, which are these. While if the nerve is in the CNS, then myelin would be made out of the oligodendrocytes. So let's take a look at what both of these cells really are. These cells are basically made out of a lot of fat, the cell membrane, and then they wrap around the axon. So Schwann cells, each one of these is one Schwann cell, and you can see the nucleus over there, and again it wraps itself around the axon, leaving these little gaps in between. These gaps are known as the nodes of Ranvier. And again, the Schwann cells are responsible for myelinating axons in the peripheral nervous system. In the CNS, we have oligodendrocytes. So if you break down the word, that means the cell with few dendrites. And you can see here that is the cell body of the oligodendrocyte with the nucleus, and all of these are branches. And you can see that this one cell is helping in the myelination of more than one axon, and more than one part of each axon. So comparing to the Schwann cell that can only wraps around only one portion of the axon, compared to your oligodendrocyte, it branches out and it enters into the myelination of several axons and several parts of the axon. And again, the gaps in between are known as the node of Ranvier. Generally speaking, myelination speeds up the propagation of the action potential or the electricity on the axon. So we kind of already talked about Schwann cells and myelin. So in the peripheral nervous system, glial cells known as Schwann cells form the myelin sheath, while in the CNS, myelin is formed by oligodendrocytes. The myelin sheath, as we mentioned, speeds up the conduction of the action potential along the axon, and that conserves the energy. Now, if you take another look at the axon, or at the neuron, you could see here again your cell body with the nucleus and all of the different organelles. So we have the rough ER, mitochondria, smooth ER, and so on. And this is where different proteins are going to be made. Some of these proteins are neurotransmitters, meaning that they allow for the transmission of information from the nerve to whatever's on the other side of the nerve. So either to another nerve, to a muscle, or to a gland, or whatever. And these chemicals are known as neurotransmitters. Again, they are made over here, packed into a vesicle, and then they have to be transported down the axon all the way to the axon terminal, and then released through exocytosis to leave the neuron. Well, these vesicles cannot just swim all the way down the axon. They are carried by different proteins. So that protein, this protein, is known as a kinesin protein, and it literally carries that secretory vesicle down the axon, away from the cell body, to the axon terminal. Again, the neurotransmitter is released by exocytosis, and then now this empty vesicle needs to go back towards the axon to be recycled. So we will fill it up again with more neurotransmitter. Going back to the cell body is carried by a different protein, and that is known as dynein protein. So let us take a look at how these little vesicles work. There's no sound to this, but here you can see the kinesin protein walking down the microtubules on the axon, carrying that vesicle all the way down to the terminal. Now, if we take a look at the other video, again, there's no audio on this either. You could see this one, this protein, because the secretory vesicle is empty, so this moves a little bit faster than the other one. You want to be familiar with your protein names. Again, so kinesin and dynein. Now, what do the neurons look, or how do we classify the neurons according to their structure? This is the one that we usually use as the default kind of neuron. Okay, these are known as multipolar neurons. They do, in fact, make up about 99% of our neurons. They're known as multipolar because you can see all of these dendrites attached to this cell body. These make up our motor neurons and what are known as interneurons. We will talk about interneurons and define them in the next slide. The second kind of neuron we want to talk about is known as a bipolar neuron. And it is known as a bipolar neuron because we've got an axon with two poles coming out of it, hence the name bipolar. These processes, one side of it is a dendrite, so the electricity would be going in this direction, while the other process is an axon, and the direction of action potential would be going away from the cell body. These bipolar neurons are found and carry special senses. So special senses are the senses that come from head and neck, like vision, smell, taste, hearing, and equilibrium. The third kind of axons we have, according to structure, are known as unipolar. And you can see here the cell body with this one extension coming out, or this one process, and then that process splits into two. A peripheral process that goes to the peripheries, so far away from the trunk, and then the central process that would enter the CNS. These unipolar cells carry general senses. So, for example, pain, touch, and pressure. So these peripheries over here would have receptors to receive the pain, touch, and pressure. It will take that sensory information towards the spinal cord, towards the cell body, and then through the central process in order to enter the spinal cord. So again, multipolar, the majority of our neurons for motor and interneurons. Bipolar are for special senses. Unipolar are for general senses. Now, these are the classification of neurons according to function. So what kind of information would each of these neurons be carrying? So you can see here, the blue area represents the CNS. This area represents the PNS, the peripheral nervous system. So let's take a look at how this is working. This right here is a cell body, and this is a unipolar cell with a peripheral process and a central process. So again, the peripheral process will be going towards, let's say, the skin, because this is general senses. Okay, and at the very terminal, we would have receptors. Let's say we're talking about pain receptors. The pain receptors will pick up that pain. The pain will propagate along the peripheral process towards the cell body, and then through the central process to enter into the CNS. Now, it meets, or it synapses. That connection between two neurons is known as a synapse, synapse, with another neuron, and this neuron is a multipolar neuron. Okay, and you can see another synapse and so on. These neurons are entirely in the CNS. As you can see, they do not leave the CNS. These neurons are known as interneurons. So interneurons connect sensory neurons to motor neurons. They connect different kinds of neurons together, and they are entirely within the CNS. In this example, the interneuron is connecting sensory to motor. Now that we've felt that pain that we just talked about, the interneuron will send an order to the motor or the efferent neuron down this multipolar nerve in order to reach a muscle, gland, or neuron, another neuron. Okay, so this axon terminal, again, will synapse with either a muscle, a gland, or a neuron. So again, by function, we have a sensory afferent, a motor efferent, and the interneurons that connect these nerves together. And to remember which one is sensory and which one is motor, there is this nice mnemonic: sensory afferent, motor efferent. Characteristics of the three classes of neurons that we just talked about. So the afferent is your sensory that transmits information into the CNS, so in the direction of the CNS from the receptors in the peripheries. And then that single process, remember we said that unipolar cell will split into a peripheral process, the peripheries in the peripheral nervous system, and the short central process, or the axon, that enters the CNS. Okay, remember we said that the unipolar neurons are for general senses, while bipolar neurons are for special senses. Now, the motor or the efferent neuron that transmits information out of the CNS to the effectors. These effectors, again, could be either a muscle, a gland, or another neuron. Your cell body are made out of, have multiple dendrites, so these are multipolar neurons. And the axon will take the information from the CNS away towards the peripheries. Your interneurons, these are integrators, and they, again, connect neurons together, and they are found entirely within the CNS. These interneurons, again, are the majority of our nerves, and these account for about 99% of neurons, and they are all multipolar. Now, what is a synapse? So remember we said that a synapse is a connection between a nerve and something else. So it could be a connection between a nerve and another nerve, as we see in this image, or a connection between a nerve and a muscle, or a nerve and a gland. So whatever the nerve connects with, again, this is known as a synaptic area. In this example, we see a connection between one nerve and another. And this right here, where they meet each other, is known as a synapse. So we have the nerve before the synapse, known as the presynaptic neuron, and the nerve after the synapse, known as the postsynaptic neuron. But you can also see here that this nerve in the middle is synapting with this third nerve. So in this area, this right here would be your second synapse. So in that scenario, this is now the presynaptic neuron, while this is your postsynaptic neuron. Okay, so one nerve can be, presynaptic in one scenario or postsynaptic in another scenario. In order to overcome that gap between the two neurons, that depends on the transmission of neurotransmitters or chemicals. Now we'll discuss the glial cells. Glial cells are the helpers of the neurons. So your main cell of the nervous system is your nerve cell or neuron, but they are helped with glial cells. Glial cells, the ones that we have in the CNS, are about 4. There are more, but the ones that we want to familiarize ourselves are 4. And before we talk about them, let us take a look at what are we looking at. Well, this is a portion of the CNS, let's say the brain, and you can see here one neuron and another neuron, and then there is a capillary, and this is the lining of the cavity that contains the CSF, or the cerebrospinal fluid. So remember that your brain and spinal cord is swimming within a subarachnoid space that is full of CSF. So that right here, these funny looking cells, line that cavity full of the CSF. Now let's take a look at all of these different cells that actually outnumber the neurons. The first one we want to discuss is this green one that is known as an astrocyte. Astrocyte means the cell that looks like a star. So you can see here what it looks like, and it extends. It has all of these different extensions where it connects the cell body to the capillary. Okay, so in the brain, in the CNS, your neurons also need oxygen just like any other cell, but it cannot directly be in contact with a blood vessel. So there is a barrier between your neurons and the blood vessels or the capillaries. Part of the barrier is made out of this astrocyte, and that barrier is known as the blood-brain barrier. So the barrier between the blood and the brain, the BBB, okay? And so through these astrocytes, nutrients will go from the capillary and reach the neuron cells. And then any waste products made by the cell body will go through the astrocytes and be dumped into the capillaries. And this blood-brain barrier is extremely important because not all medications are able to reach our brain cells, okay? The meds that we need in order to affect the neurons in the CNS have to have the ability to cross this blood-brain barrier. The second nerve we want to introduce are these oligodendrocytes, and we actually already discussed those, and those make the myelin sheath within the CNS. We also have these microglial cells. These are cells of the immune system. They are modified macrophages, and they have the ability to identify and eat up through phagocytosis any foreign bodies trying to invade the CNS. The fourth and the last kind of cells we are going to introduce here are the ependymal cells, and these line the cavities of the CNS, whether it's in the brain or in the spinal cord, and they are responsible for the making and maintenance of the CSF. So they secrete CSF. Now here you can see the glial cells in writing. Okay, I kind of prefer to use the image first and then move on to the writing. So the astrocytes, those, as we mentioned, are part of the blood-brain barrier. So BBB here does not stand for Better Business Bureau. It is the blood-brain barrier. And they help regulate the composition of the extracellular fluid in the CNS. So again, they are going to take the nutrients towards your neuron, get rid of waste products, make sure that the correct number of potassium, the right amount of potassium is found within the extracellular fluid, and help in the transmission of neurotransmitters. The microglia, these are specialized forms of macrophages, and they perform the function of the immune system in the CNS. Microglia also have a function in remodeling and plasticity. So we are going to talk about the brain, the ability of the CNS to make new connections as we learn more things. Okay, that process is known as remodeling and plasticity. Ependymal cells, those line the fluid-filled cavities found in the brain and the spinal cord, and they help regulate the production and the flow of CSF. The oligodendrocytes, those form the myelin sheath within your CNS. Now, what about the development of the nervous system? And we're talking in fetal life. So in embryo, it starts off with a stem cell, just like any other system within our body. So we've got a stem cell, and that stem cell will be exposed to what are known as neurotrophic growth factors. So growth factors that are specific for the growth of neurons. And that is why these specific stem cells are going to develop either into a neuron or the helpers, the glial cells. So you can see here, this that stem cell was exposed to those neurotrophic growth factors and is developed into a cell body and an axon. So how does that axon know where to go? Okay, so this axon develops at the very end a growth cone. That growth cone is going to be guided by also these neurotrophic growth factors and other chemicals to find out their specific route. So is this going to end up going to the biceps muscle, for example, or is it going to end up going to the quadriceps muscle, which is in the lower limb? Okay, so it'll follow those chemicals to find out what the final target is. So which route the axon follows depends, as I said, on these neurotrophic growth factors and other chemicals, for example, known as CAMs, or the cell adhesion molecules. And these are found in the surrounding fluid, or the fluid surrounding that growing neuron. So it's found in the extracellular fluid surrounding the growth cone and its distant target. So these again are kind of like guides to figure out where that axon will be going to. The growth cone will keep on growing, guided by these factors until it reaches its target and it starts to form a synapse. Okay? During these very early stages of neural development in utero, these factors can be affected by alcohol and other drugs, radiation, maternal malnutrition, and viruses can all lead to permanent damage to the development of the fetal nervous system. Now, throughout our lifespan, the brain has the ability to modify the structure and function, and that ability to modify is known as plasticity. So, for example, that baby that was born that does not know how to read or write or count will eventually learn these skills and make connections, increase the number and the branching out of the dendrites, and that process is known as plasticity. So we do not make new neurons. Okay, so after birth, after the CNS, if we damage the neurons, it is almost impossible to regenerate them. But we continuously have the ability to increase the connection between neurons, again, to increase the number of dendrites, the branching out, and that modification process is known as plasticity. So you want to think again about this kid, this 1-year-old that isn't able to recognize letters or numbers. They will learn how to do that. So when they see a letter, they would recognize the letter, and then they will learn how to pronounce that letter. So there has to be connections between the eye that sees and the muscles of the tongue, or the other muscles that will pronounce that letter. These connections that are made, again, is known as plasticity. Okay, so we keep that ability. It's obviously much malleable. It's more malleable during childhood, but we keep that ability, and in order to keep that as we age, exercise is crucial. Also engaging in cognitively challenging activities. So, for example, doing crossword puzzles, maybe doing math, trying to solve math problems and so on. So things that challenge your brain, cognitively challenging activities, stimulate that plasticity. And that is something we usually tell kids, especially when they're younger. Don't take that 4-month break of summer vacation and not do anything at all. Okay, so you want to make sure that you're exercising during break, and also maybe taking 5, 10, 15 minutes out of your day to do maybe math puzzles, math problems, reading, and so on, just to make sure that you're always stimulating your brain. Now, we kind of touched upon regeneration in the CNS, okay? So we said that if we damage neurons in the CNS, so in the brain or spinal cord, if you damage those cell bodies, it is almost impossible for them to regenerate. Okay, and that is why you will find that stroke patients or injuries to the spinal cord are extremely hard to regenerate. And most of the time they do not regenerate at all. What happens though if we injure the peripheral nervous system? So we will take a look at that. Again, it depends on where the injury is. So if the injury is to the cell body, that is a problem, okay? Because again, The cell bodies have the nuclei. This is where the ATP is made, the neurotransmitters are made. This is where the nutrients are coming from. Nutrients come from your cell body and go down the axon. So if you damage that source, then regeneration is not going to occur. But if the injury is in the axon, that can regenerate. And that's what we will be discussing in this slide. So you can see here, this is the area of injury. That's the site of injury. Anything proximal to that injury is still getting oxygen and nutrients from the cell body. So this part of the axon survives while this part dies off. Okay, so we are going to lose the distal portion of the axon. And notice that I said the axon, not the Schwann cells. These Schwann cells receive their nutrients from a different source. So their survival is not dependent on the cell body. But we will definitely lose the axon. We will lose these connections between the axon and that skeletal muscle. But you see here that the Schwann cells survive, and they perform what kind of like a tunnel, so that this peripheral part is able to grow and regenerate, using that tunnel as a guide in order to reconnect with that same point at the skeletal muscle. Okay, so at some point, we can reestablish these connections. It takes a long, very long time though. It's about 1 millimeter per day, which is a very slow process. So if, let's say, somebody injured the nerve, let's say at the elbow, okay, and that nerve was supposed to innervate a muscle in the hand. It'll take months, maybe up to a year or so, for that whole nerve to regenerate. So that is something that can be very frustrating for the patient. But again, you want to explain to them that it is a slow process and that these connections will eventually get back to normal. And it's all about patience, which again is a very hard thing to do when you are dependent on your small muscles of your hand. So the injury, again, if the axons are severed or if they're cut, they do have the ability to regenerate and restore the previous function as long as the cell body is intact. Okay, so remember again, your cell body is where you get oxygen and nutrients and so on. So after the injury, again, if it's in the axon, the part of the axon that is connected to the cell body will remain healthy. The part that is distal to the injury will die off. The axon that is still healthy will also grow a growth cone, and then it will go through that tunnel that is established by those Schwann cells. Okay, again, as I mentioned, it is a very slow process, about 1 millimeter per day, and it is guided by those Schwann cells. So it is really, the regeneration of the neuron is really dependent on the survival of those Schwann cells. Spinal nerve injuries, okay, those tend to crush as opposed to cut the nerve. Okay, so somebody had spinal nerve injuries, for example, a disc injury, okay, disc bulging. Those tend to crush the nerve and not cut it. Okay, when you crush the nerve, that can lead to the death of the Schwann cells. Okay, that death of the Schwann cell is known as apoptosis. And remember, the regeneration process really depends on the survival of that myelin sheath. So if the myelin sheath does not survive, the regeneration and the full recovery of the nerve might not occur. Okay, so in the cases of spinal nerve injuries where we again crushed those the glial cells, where they became apoptotic, that might lead to the absence of any kind of significant regeneration of the axons. Since the injury occurred in the spinal cord, very close to the spinal cord, these are no longer Schwann cells, those are oligodendrocytes. So, what is science trying to do about this? Are we just going to live with the fact that we cannot regenerate our CNS neurons? Keep in mind that in utero, the CNS did in fact grow in the absence of these oligodendrocytes and the glial cells. We had a growth cone, we had neurotrophic growth factors. So why is it that we cannot recreate that environment so that the neurons can regenerate if an injury occurs? And that's what the science is trying to do now. So we are trying to implant undifferentiated stem cells, recreate what happened in utero. Okay, so create tubes. And again, the axons have no idea. Okay, they need direction. So we want to create these tubes, maybe try to prevent apoptosis of the myelin sheath by supplying these neurotrophic factors. And maybe, just maybe, the neurons will have the ability now to regenerate. So again, this is all research that is in progress. So at this point, I'm going to stop this very first portion of Chapter 6, and we will continue later.